Hello fellow biologists, I'm here to talk to you guys about meiosis. To have a general understanding, meiosis is a process where a single diploid cell divides twice to produce 4 haploid cells containing half the original amount of genetic information. The main purpose is to produce gametes or sex cells such as sperm and egg cells to be used in sexual reproduction. Meiosis can be split into 2 main parts, meiosis 1 and 2. In each part is a few phases. So, let's begin. Let's go! Before meiosis 1 can begin, a process known as interphase occurs. As you can see, we have a diploid cell containing 6 total chromosomes in its nuclear membrane, 3 from dad and 3 from mom, specified by their color. During interphase, each chromosome goes through DNA replication. After DNA replication, each chromosome now consists of 2 identical chromatids joined at the centromere or center. These 2 chromatids are called sister chromatids. By the end of interphase, you can see that each of the 6 chromosomes have undergone DNA replication, and now meiosis 1 begins. In meiosis 1, the first stage is prophase 1. In prophase 1, several processes occur. First, the nuclear membrane dissipates, and the spindle fiber, a crucial part in later stages, is formed. The replicative chromosomes begin to pair with their corresponding homologous chromosome, meaning mom and dad pair up, to form a homologous pair, which is also called a tetrad. In prophase 1, when the homologous chromosomes are paired up, crossing over may occur, which is where bits and pieces of the homologous chromosome are exchanged, which causes genetic variation. Yay! Next comes metaphase 1. In this phase, the paired homologous chromosomes are lined up across the center of the cell and the spindle fibers have fully formed. Then there's anaphase 1. This is where the spindle fibers pull the chromosomes from each homologous pair toward opposite ends of the cell. When anaphase 1 is complete, telophase 1 and cytokinesis occur. In telophase 1, a nuclear membrane is reformed around each chromosome cluster at each end of the cell. In a cytokinesis, 2 new cells are formed. And that concludes meiosis 1. As you can see, the original diploid cell is split into 2 haploid daughter cells with 3 replicated chromosomes or 3 copies of half the genetic information. Now each cell begins to undergo meiosis 2. Thought we were done? Nope! Although meiosis 2 is quite similar to meiosis 1, there is no second interphase before meiosis 1, meaning there is no DNA replication that takes place. Instead, each cell goes straight to prophase 2. As the cells enter prophase 2, once again, their nuclear membranes dissipate and the spindle fibers begin to form. The chromosomes, each consisting of 2 chromatids, become visible. They do not pair up to form tetrads because the homologous pairs were already split up in meiosis 1. In metaphase 2, the spindle fibers fully form and the chromosomes line up in the center of each cell. Anaphase 2 then occurs, where the spindle fibers separate the paired sister chromatids of each chromosome to opposite ends of the cell. Then, telophase 2 and cytokinesis occur, where a nuclear membrane is reformed around each chromosome cluster and each of the 2 cells split to form 2 new cells for a total of 4 haploid daughter cells. Now that is the process of meiosis. In essence, meiosis is crucial to the continuation of sexually reproducing species. It ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes and produces genetic variation by way of crossing over. But wait, what is the relationship between Mendel's model of inheritance and meiosis? Well, it's simple. Meiosis proves Mendel's model of inheritance. Mendel created a series of certain laws, principles, and claims that can all be proved by meiosis. Mendel's first claim is that alternate versions of genes account for variations in characteristics. In meiosis, the crossing over that occurs during prophase 1 is accountable for genetic variation. Another claim by Mendel is the law of dominance, which states that a dominant allele masks the traits of a recessive allele and determines the characteristics of a specific gene. In meiosis, when gametes are formed, the dominant allele determines the phenotype of a certain gene and will be expressed in the offspring. The same is true when alleles cross over in prophase 1. Mendel also created the law of segregation, which states that alleles for a gene separate during gamete or reproductive cell formation. This proves true in meiosis 1, where in anaphase 1 and 2, homologous pairs of chromosomes are separated and sister chromatids are separated, respectively. Mendel's final claim is the law of independent assortment, which states that each pair of alleles segregate independently during gamete formation. In meiosis, alleles are sorted randomly and independently in crossing over, so there will be no definite combination of alleles in gamete formation. This has been the process of meiosis. Thanks for watching.